So, well, Christina said it already. Thank you very much, everyone, for being here. This is the fourth, if I'm not wrong, this is the fourth masterclass of this series. These masterclasses are part of the level 3 of the EPIC project. The EPIC project is a European project that the goal of this project is to increase the capacity, the knowledge in identification of butterflies through courses. In this case, this is the fourth masterclass of this level 3. The expert we have today is 𝘍𝘠𝘍𝘠𝘍𝘠𝘍𝘠𝘍𝘠. It's an expert in butterflies. Concretely, is expert on Lysandra species. Miguel has been a zoology professor at the Universidad Autónoma of Madrid in Spain until last year when he retired, but he's still quite active doing different things like this talk. His research was mainly focused on Iberian butterflies with emphasis on Lysandra species. For instance, his PhD was about Lysandra species and conservation. He has published more than 100 papers between book chapters, journal papers, and other kind of contributions. He has been in the European Butterfly Atlases and Red Books, and he has also coordinated Spanish Atlas, Butterfly Atlas, and Butterfly Red Book, and that this has been a recent work that has been completed and will be published soon. He has been also the chairman of Butterfly Conservation Europe for some years, and also the coordinator and the driving force to establish the Spanish Butterfly Monitoring Scheme. So I think that's all from our part. Miguel, thank you very much for being here today, and the floor is yours.
Okay, thank you very much, Juan Pablo, and thank you very much to everybody listening. Well, this talk will be quite long, so be patient. We will have a short break after we talk about the Polyommatus. And well, I just wanted to say that most of the photos, most of the pictures you will going to see are from myself and from Juan Pablo, and I will also be using some pictures taken from Fauna Ibérica, this monograph on about the Spanish and Portuguese butterflies. So let's start. We will talk about these 5 genera: genus Polyommatus, Lysandra, Palaeophilotes. This is a new name from what was previously considered 𝘬𝘪𝘍𝑜𝑝hilotes. So sorry about this, but taxonomy is in continuous change. The 4th genus would be Agriades, and then Arithia. For all these genera, we will be producing a list of species. Then we will talk about diversity and the main characteristics of the genus. And then we will give some hints on species identification, habitat and ecology, distribution of the species, unique traits, and we'll compare each species with similar ones. So these are the contents. So for start, I will point out things that I will be talking about all along my talk. And you must keep an eye when you are identifying Polyommatus or other Polyommatine species, all these characters. First of all, the fringes. Sorry, I will change the marker. So the fringes are these scales on the border of the wings. You will also have to take a look to the cell spot in the forewing underside here. So, for example, this picture has these spots. Also, the position of spots in the hindwing underside, these spots here, is important. Also, the present shape and color of orange luminous, these spots here, that are orange. Also, the other character is the shade of blue in the species that have blue males. So, for example, this is a male, and the shade of blue is important for identification. And also the presence of this white triangle here that can also be a band in the hindwing underside. So this is also important for identification of some of the species. So don't mind about all this list, but because we will be treating with a lot of examples all these features. Sorry. Well, we start with Polyommatus. Polyommatus is a large genus, and in Europe you have all these species, and we will be considering the ones that are marked in bold, so quite a lot of them. So let's start. We will treat them not all of them together, but in different groups. So for a start, I will give some ideas about the diversity and morphology of Polyommatus. There are 25 species in Europe. Several are endemic with very restricted distributions, particularly endemic to the Iberian, Italian, and Balkan Peninsula. So the peninsulas are important for this genus. They are mostly distributed throughout the Mediterranean countries. So the Mediterranean is a red spot for the diversity of this genus. They usually have strong sexual dimorphisms with males blue usually and females brown, but some anomalous blues, for example, are in these species, the males are brown, so that's an exception. Fringes are not checkered, so we will see this in a lot of examples. And the blue shade of males can be diagnostic, but this is a difficult trait because there are so many species, and of course, the shades of blue are quite diverse. And another important thing is that orange markings and the position of dots are also important diagnostic features. So we will start just having a look to four pictures. You see that the three of them are Polyommatus, and the fourth is a Lysandra. So it's quite similar to the Polyommatus. We will see the differences when we study the Lysandra species. We will start with this group, the Dorylas group. These are beautiful butterflies with a very, very nice blue in the upper side of the males. Now, all the species from this group have a white marginal underside here and this is particularly specific in the apex of the wings. So here you can see a white margin that is typical in the three species. You can see it. Now, the other character, important character for this species is the shade of blue. For nivescens, it's shiny pale blue. Well, it's difficult to describe these blues, but you can see it's quite distinctive. And in the other two species, it is turquoise blue, so it's brighter in some way. The other thing you have to have a look at is the orange markings that are reduced in nivescens and are quite conspicuous, quite contrasted in Dorylas, and also quite showy, but not as conspicuous as in Dorylas in Polyommatus golgus. these two species are quite similar, but they don't fly together. So geography is also an important feature. And so we will see these species one by one. First, Nivestans. This is lives in mountains in the eastern half of the Iberian Peninsula, and it's a habitat specialist using calcareous areas. Also, it's typically rocky areas or open areas in different kinds of woodlands. The larval food plant is 𝘗𝘕𝑽𝑡𝑗𝑃, Antilles vulneraria. You see in Spain, typically 𝘗𝘕𝑽𝑡𝑗𝑃 is red or pink, but as you might know, in other places of Europe, Antilles vulneraria is yellow. So, and it's also so in some parts of northern Spain. So, but this is a typical aspect of the plant in most of the species distribution. Adults fly from April to October, but well, when I talk about phenology, of course, it's quite difficult to find specimens in April and also in October, but you can. But you mainly will find specimens in July. It is endemic to eastern Spain, so you cannot find it in other areas in Europe. And well, as I told you previously, males are easy to separate due to this unique shiny pale blue upper side. The next species is well, I put these two species together because they are quite similar, although they live in completely different habitats. They can live in dry flower-rich slopes in calcareous grasslands, or this is the case of Dorylas, or open patches in dwarf scrub and grassy vegetation growing between schist or limestone rocks in the south of Spain at high altitude areas. The larval food plant is again 𝘗𝘕𝑽𝑡𝑗𝑃, Antilles vulneraria, and again adults fly well from beginning in the spring, but mainly during the summer. The distribution is quite different. Dorylas is a widespread species, mainly Central and Southern Europe, but Golgus is restricted to the mountains of southeast Spain, so they never fly together. And of course, although they are quite similar, you cannot find them in the same places. So if you are here in the southeast, then you will get Golgus. This is a female, and otherwise in other places you will find Dorylas. The next group is formed by these three species. Now, these three species lack the cell spot in the forewing underside. So you see there's no spot here, no spot here, no spot here. And of course, it's different from the previous species, of course, because of this white band in the margin. Now, the first species, 𝘗𝑰𝘭𝘺𝑽𝑡𝑗𝑃, it has prominent V-shaped black marks here. This V-shaped marks are quite typical, and also it's got quite big points, big marks in the underside. The next species, 𝘧𝑟𝑠𝑖𝑡𝑒s, has parabolic and conspicuous orange lunules. This is a characteristic for this species. And then the third species lacks this triangular white mark. You see here, white mark, but Amandus doesn't have a white mark. So this is typical for this species. And also, the veins are marked in the upper side more than in the other two species. So these two characters, the veins and the absence of white triangular mark are characteristic for Amanthus. And just remember, the three species lack this cell spot in the forewing. Let's have a look to each species. 𝘬𝘰𝘭𝘨𝘦 is a widespread species in Southern Europe, particularly for the three peninsulas in the south. It lives in open, stony, dry areas, grasslands, and also low scrub with calcareous, again, a calcareous substrate. Its larval footplants are several, but mainly astragalus and Oxytropis. Oxytropis is quite similar to astragalus. Here you have an example, Astragalus montpesulanus. That is one of the typical footplants, larval footplants in Spain. And adults fly, well, mainly at the beginning of the summer, I would say. And then the distribution is, this distribution is only for Europe, but it also flies in Northern Africa, Morocco. So remember, the underside has big spots and these V-shaped black lunules that are characteristic for this species. The next species is Thersites. It lives in a wide variety of habitats. You see, also the distribution is quite wide. It reaches China. The larval footplants are several legumes. For example, this Sonobrichis is one of the typical footplants, but it can use many other ones. And it has two generations, and this is different from the previous species. This, for example, 𝘬𝘰𝘭𝘨𝘦 has only one generation, but Thersites has two generations. As I told you, the distribution is quite large. And then the main characteristics is that, of course, it hasn't got a cell spot, and then the orange markings that are large and parabolic, as you can see in this picture. The third species of this group is Amanthus. It's more a wet grassland species, or more a habitat specialist. Its larval footplants are again legumes, but mainly are vetches of the genus Vitia. You have here the flowers of this plant. And adults fly in one generation from May to September, but mainly during the summer. The distribution of this species is again quite large. It's widespread in several places, even from North, Central, and Southern Europe. And remember, the underside has no triangular mark, no triangular white mark, and the upper side has white veins. The upper side has marked veins, as I told you previously. So this is the second group. The third group is the Anomalous Blues. This is an interesting group. In Southwest Europe, it's got 5 species, so this is a larger group. Now we must first have a look to the males. Have a furry patch on the base of the forewings, and these are androconia. These are scales that the male uses to produce chemicals that attract the females. Another character is that there is a conspicuous white band that is absent in some species or areas, but this band is quite typical. And then another character is that the marginal spots are reduced with no black or orange markings, while the postdiscal spots, these spots are quite conspicuous. So these characters are common for all the species of the group. In Southwest Europe, we get these 5 species. You can see, well, this is a difficult group. First, we have the species with brown males. Sorry, I put the males and the females in different, this is a female, this is a male, this is a male, this is a female, but however, the males here and here of these three species are brown. Now, what's the difference between these species? Reparty has this white band, but not always. In Catalonia, it hasn't got a white band. We will see that the distributions don't overlap. So we will see that later. And Fabracea and Violetae usually don't have this white band, but Violetae sometimes has it. So then again, this is a tricky character. With the blue males, it's easier because Fulgens has got a pale blue upper side that you can see here. And Damon has got a bright blue upper side with a black or a dark margin, and also the veins are quite marked. Now, the females of these species are extremely difficult to tell apart. The only exception is the females of Damon that don't have. You cannot see the veins in the upper side. You can see the veins here. You can see the veins here, and you can see the veins here, but not in Damon. So Damon is the only species that you can more or less identify easily with the females. The males are easier because of these blue or brown characters. Now let's have a look to the ecology of all these species is quite similar. They always live in calcareous areas in a wide range of open habitats and grasslands. The larval food plant is always Synthron, Onobrichis, as I told you previously, for one of the Polyommatus species. And adults fly in the summer. That's all we can say. And the distribution, the distribution is also distinctive. So you must, when trying to identify these species, you have to have a look to the place where you are. There are three endemic species. These three, the Fulgens is a northern species in the Iberian Peninsula. Fabracea occupies a central area. This is the Iberian mountain system, and Violetae is a southern species. So they don't overlap. These three species always fly in different areas. The widespread species follow, and but I must say that reliable identification may need DNA or chromosome studies. But as I told you, the distribution helps. So distribution, as we will see, is quite important. Now, with the widespread species, this is Reparty that can be found in the three peninsulas, and Damon is also a Central and Southern European species. For example, in Spain, you can find Damon in the north and in the center. So in the center, as I showed you, you can find Fabracea, Reparty, and Damon. Damon is quite easy because of the blue males. Now, what happens with Reparty and Fabracea that are quite similar? Now, Reparty has this white band here in the center, and Fabracea doesn't. So when you're here in the Iberian mountain system, if you find a specimen with a white band, it will be Reparty. But if you find a specimen without a white band, then it will be Fabracea. And that's the only clue we have to identify these species. But as always, there is an exception. Reparty has a white band everywhere, but in Catalonia, you see specimens without a white band. So they are quite similar to Fabracea, but they are not Fabracea. Okay, so they are repartee without a white band, but this is only in Catalonia. So otherwise, repartee has also this white band. Well, so at least for Spain, this more or less works, although it's quite difficult. But it's the only clue we have to identify species. Now, the next group is the Polyommatus icarus group. These are species that are similar to Polyommatus icarus. The common blue Polyommatus eros is a mountain species. You see the blue is more shiny than in Icarus, that has a violet blue in the males. In the underside, you find cell spots in all these species. So this is distinctive. All this Polyommatus icarus group, they all have these cell spots. And to identify these two different groups, Eros has usually a darker underside and reduced orange lunules, while Icarus and Selena have a paler underside and then quite prominent orange markings. Now, how can we tell apart Icarus from Selena? Well, forget about this. You really need a DNA study to separate these two species. But however, we will see that their distribution overlaps in a very narrow area. So in most cases, the distribution can help. Okay, but otherwise, you cannot tell them apart. They are very similar, and the supposed differences are really variable as well. So this is difficult. Now let's have a look to the species. Eros is a mountain species. It lives in rocky grasslands at high altitude areas. The larval food plants are several legumes, but particularly Oxytropis, that is a genus that is similar to Astragalus. You can see here an example, and it's got a one generation from June to September, but mainly if you are looking for this species, you go in July, the beginning of August, mainly. And well, its distribution, you can see that it lives in mountains in several areas in Europe, particularly the Pyrenees, the Alps, and then the Balkans, the Apennines. This species is similar to Icarus, but the male upper side is quite different. Now, the next, of course, I grouped Icarus and Selena because they are so similar. They occupy a wide variety of grasslands and open areas. And also you can find them in alfalfa crops. So this is one of the few species that can live in such kind of habitat. The larval food plants are several Fabaceae, but for example, Lotus, Medicago, and Trifolium are quite typical larval food plants. This is Lotus corniculatus, one of the typical food plants of the larvae. And well, these species are polyvoltine, so you can find them throughout the year, particularly in southern areas. So having 2 or 3 generations, and in southern places you can find them from all along the year. Now, the distribution is quite distinctive. Selena is, I didn't show you the map because the limit of the two species is difficult to establish, but Selena is in the south. So, for example, if you're in Andalusia or you're in the Balearic Islands or in Sardinia, or in North Africa, or in the Canary Islands, what you will have is Selena. In the rest of Europe and further beyond, what you will have is Icarus. So here, geography is useful, but there's a wide, narrow strip in the center of Spain in which you will never be sure about it. So to be sure, you can only rely on DNA studies. So that's where the problem with these two species. Now, to end up with Polyommatus, we have this beautiful species, Polyommatus daphnis. This is quite distinctive. That's why I separate it from all the rest of the genus. Now, in this species, males are blue, females can be blue or brown. It depends on the specimen. So you can find these two morphotypes. In the underside, there's no orange lunules. So this is another character. But the main character for this species is this undulated margin. So it is more evident in the females than in the males, but the males also have this undulated margin that is typical for the species. Okay, so let's have a look to, and of course I forgot, but this species doesn't have a cell spot. So let's have a look to the features of this species. It lives in dry and stony grasslands and open scrub, again, calcareous, or in this case also it can also use gypsum substrates. The larval food plant is typically astragalus. This is, for example, Astragalus monspesulanus, one of the typical food plants of the species, but it can also use other legumes, so it's not monophagous as other examples from the genus. Adults fly from June to September, mainly during the summer, in only one generation, so this is also typical. The distribution, as you can see, is from, Central and Southern Europe. It also reaches Asia and, Iran, where the bombs are now falling. Males are, the features for this species, as I told you, males are blue, but sexes have this undulated margin, and they also lack these orange lunules that are typical for all the species. So we have already finished with the Polyommatus. So this is more or less in the middle of my talk. So let's first answer questions, and then let's have a break after this.
Okay. So, we don't have questions, Miguel. That's not bad news. I think people are still processing. I think it's quite clear what you are been explaining. We can give some minutes for that break, 5 minutes. Maybe we have some questions when everyone is back.
Well, if any questions arise from this genus later, I can, of course.
Sadly, we give time. But I think it's pretty clear. Maybe only the Anomalous Blues are the most difficult.
Yes, but they are difficult for everybody. So you have to bear in mind that for this species, it is very difficult. You have to know where you are, and the characters are sometimes variable. So yes.
Miguel?
Yes?
I have a question about the Anomalous Blues.
Yeah.
They are kind of specialized, no? So maybe they are in high mountains or...
Well, not really high. I would say medium mountains. There are, but not... They are using a wide range of grasslands. So the habitat is not particularly... Of course, grasslands and calcareous. But the habitat is not particularly different, I would say, from other species. You will find them in areas where most lysanids will be flying. So that's not particularly different, I would say.
Okay.
So and if you see the distribution, it's always mountains, but not high altitude.
Not super high altitude. Okay.
Yeah, because for example, if you see here in the Pyrenees, they don't really fly in the highest areas in the Pyrenees, and it's the same in Sierra Nevada. They don't fly in the highest peaks. So it's well, it's how it is. It's more medium altitude.
Okay, thank you.
Okay.
I think I'm back already. You were talking about the anomalous blues again.
Yeah, I had a question about that. So
good. I think people they don't realize how difficult is this when you are in the field. I mean, most of the participants here of the assistants, they have a lot of experience in the field, but when you start seeing this experience in the field, and then you need then to go to another.
Well, however, if you follow the instructions, you can more or less be sure in, well, say, most of the specimens. So,
exactly.
Of course, there's a small percentage in which you need another analysis. So yes, exactly. This is. So can we?
Yes, I think we can
carry on.
Yeah.
All right. Thanks a lot. Now we will. The next genus is Lysandra, or however you pronounce that. It's got 6 species in Europe, and 5 of them live in the southwest, and 1 is just from southeast Europe. So for example, if you come to the Iberian Peninsula, you can see 5 of them. There are some endemic species, some widespread species. We will see that later. Now here you have the 5 species that you can find in Southwest Europe. First of all, I must say that this is a difficult group. Males are quite easy to identify because of their different blue shades, but females are impossible. So if 2 species are flying together, the females are very difficult to identify. But you can have a clue with geography and some other ideas. You can have a clue about the species you are dealing with. You can see here that, first of all, how can you separate Lysandra from Polyommatus? The key feature is these checkered fringes. So you can see all the species have these checkered fringes, and then they all have spots in the cell. Here, these spots, you see them in every species. Well, this is all these characters are variable. However, there's a spot here, but this is a not very typical specimen. And then it's important to bear in mind the blue, the shade of blue for the males. I already said that the females are hopeless, so when you are in the field, it's better to look at the males. Also, some species are quite variable. For example, Corydon has this female variant that is quite typical. They also hybridize, so you can find hybrids between, for example, Belargus and albicans or hispana. There are hybrids between all the species. The reason why this group is so difficult is that they're evolving at the moment. So in some species you find quite nice different species because they have already separated from the neighboring species. But with these Lysandras, they are, in this moment we are living, they are just evolving. So probably 10,000 years later, they will produce quite different species, but at the moment they don't. So we have to. You will see. However, we have some clues. Don't be pessimistic about this. Now, I will group all the species because they have similar ecological features. They live in upper grasslands and low scrub. They are always in calcareous areas. Sometimes you can find some populations in acidic rocks, but that's an exception. They usually always live in calcareous areas. Larval food plants are horseshoe bedges for all the species. So genus 𝘨𝘭𝘨𝘏, you can see here an example. And they can have two spring and summer generations, like in Belargus or in Hispana, or only one summer generation, like the other three species. Okay, so that's another thing you must take into account. And the distribution is restricted for these three species. We will see later. And there are also two widespread species, Belargus and Corydon. And as I told you, this is a difficult group, and sometimes DNA or chromosome numbers are necessary for reliable identification of the species. Now let's have a look to the distribution, because this is important. First of all, Celestissima, as I showed you in this picture, is restricted to the center of the Iberian Peninsula, to the Iberian mountain system mainly. Albicans and Hispana have quite a restricted distribution. These maps come from the IUCN, so the brown areas here are the maps from the IUCN, but they are not correct, I would say, because, for example, Hispana lives here as well, and Albicans doesn't live here. So be careful with the maps, because some maps that are published here and there are not really reliable, and that's another drawback, I would say, important thing to take into account. Corydon has a widespread distribution, the same as Belargus. I didn't show you Belargus because Belargus is also a widespread species. And Celestissima, as I told you, is restricted to this area. Corydon doesn't reach this area where Celestissima lives, so Corydon and Celestissima do not live together. So this is an important thing. Corydon is more a northern species in the Iberian Peninsula, and Celestissima is restricted to this area. Now, how can we deal with these difficult distributions? We did some exercise for the Spanish butterfly atlas, and you will see here that it's also quite difficult to rely on distribution if you are dealing with these two difficult species, difficult to separate from each other. So albicans is for the violet dots, and Hispana for the green dots. And of course, if you're in Catalonia, it's Hispana. If you're in most of Castilla, most of the center and northwest Spain, it will be albicans. But if you are in Andalusia or here in the Iberian mountain system, you can have both species. Typically, albicans is a western species, and Hispana eastern species. But of course, if you're in places like that, like this here, you cannot tell precisely which species you will find in a precise place. So you must then rely on chromosome numbers, DNA studies. And the third thing is that is important here, but I didn't show you this because we are talking all the time about adults, but albicans and Hispana eggs are quite different. So if you have the eggs for the species, then you can more or less identify them precisely. But of course, getting the eggs from the species is quite difficult. So you usually deal with the adults. So, well, you see, we can more or less be sure about these species in most of the cases, but in some areas or some specimens, it is quite difficult. Now, the next genus is Palaeophilotes. I already told you that the name of the genus has changed in the last years.
Miguel, sorry.
Yeah.
Someone is asking about Portugal. You have shown the maps. Any of these species we have mentioned of the Lysandras occur in Portugal. It's not that you don't want to show Portugal, it's that most of these species, they have become a species in areas that are not in the west of the Iberian Peninsula.
Yeah, I think Belargus is you can find Belargus in Portugal, but I don't think the other species can live in Portugal. I'm not sure about Corydon here. Corydon lives in Galicia, and probably, sorry, I don't have the detailed map for Corydon in the Iberian Peninsula, but yes, in Portugal, you don't have the difficult species. That's that's a good news, I would say. Yeah. So, but you certainly have Belargus, that is one of the widespread species. All right.
Okay.
Now let's go to Palaeophilotes. As I told you, the proper genus name should now be Palaeophilotes. This is supported by a lot of evidence. So we must now adopt this name as valid. There's 4 species in Europe, and we will only be talking about these 3 species. Wickrama is more a eastern species. Here we have the 3 species we are going to deal with. Now, all species from Palaeophilotes have checkered fringes. You see it here in all the species. So this is one of the typical characters. Males are blue, although sometimes it's a very dark blue, but they are all blue. And also they have reduced orange lunules, although you can find orange lunules in some species like baton. They are usually reduced, only found in the hind wing. And another character that is quite obvious is that the markings in the underside are quite conspicuous. Now, how can we tell apart all these 3 species? First, have a look to the blue. Panoptes and baton have quite what I would say is a sky blue, so quite light blue, while Abencerragus has a very dark blue. Another character is about the fringes. The fringes are T-shaped in Abencerragus, but they are just square in both baton and panoptes. Well, this character is quite a good one, but normally in 3 or 4 days after the butterfly emerges, the fringes will go away, so you won't have this character available. So then you must rely on genitalia to separate these 2 species, Abencerragus and panoptes. Baton is typical because of these orange lunules. So if you have orange lunules, it's baton. If you don't, it's Abencerragus or panoptes. And then you look at the fringes that are T-shaped in Abencerragus, square in panoptes. And if you don't have the fringes because the specimen is worn, then you have to extract the genitalia, and you see the valves of the genitalia are quite different. So this is a very, very useful character. And of course, you have to take the specimen, take it to the lab, and of course, extract the genitalia. So it's quite complicated again. We will see that in some areas you only get a Panoptes and not a Bensaragus, so that can probably also help. Let's have a look to these species. First of all, Abencerragus. It lives in grasslands or low scrub and clearings among Quercus or Pinus woodlands. Typically, you are looking for this food plant, and this is a habitat specialist, and the food plant must always be present. This Cleonia lusitanica must be present if you are looking for a Abencerragus. And if you don't find a food plant, it is quite typical that you will have one of the other species, particularly Panoptes. So this is one important thing to look for the food plant in this case. Adults are spring flyers. They fly from March to July. And the distribution, as you can see, well, they also fly in North Africa, but in the Iberian Peninsula, they only fly in southern areas. So if you're in the north or in the northeast, you will have only Panoptes or Button. Okay, so Abencerragus is only, well, there's a population here in Castilla that is tricky, but however, the habitat is always quite different with this food plant. Okay, let's have a look to the other two species that are quite close together. They are difficult to tell apart. The first one, well, they live in grasslands or low scrub, and you need the presence of several species of Thymus. The larval food plants are Thymus or Cuscuta. Cuscuta is a parasite for Thymus. So if the food plant is Cuscuta, you also need to have Thymus in the habitat. Okay, and the adults are again mainly spring, but in Button has two generations, so you can also find Button in the first generation or the second, that is in August. Now the distribution is quite different in these two species. Button is more widespread, so Central and Southern Europe, and in the Iberian Peninsula it only occupies I would say a narrow strip in the north and also Galicia reaching Portugal, while Panoptes doesn't live in those areas. No, well, there's probably this is not a correct record. So if you're in the Cantabrian coast or in Galicia, you can be more or less sure that you're with Button. Otherwise, it will be Panoptes. Okay, so well, this is of course there is a contact area in which this is difficult. For example, in the Pyrenees, Button lives in higher areas, while Panoptes lives in lower Mediterranean areas. So you can more or less, with the habitat, you can be sure if you have one or the other species. The food plants, well, this is, for example, a typical food plant for Button, Thymus serpyllum, while Panoptes uses more Mediterranean Thymus species. Now, have a look to the orange lunules that are present in Button, but not in Panoptes. And this is tricky because some Panoptes do have orange lunules, but they are usually very, very pale, very small, very tiny. So, well, that is more or less the only clue you have to separate both species if you are in the contact zone of the two species. Now, the next genus is Agriades. Agriades is a mountain or northern genus. It's got 7 species, and we will only be talking in Southwest Europe. We will only be talking about 3 species. All of them live in mountains. So the rest of the distribution of the genus is Scandinavia. These 3 species are Well, all the species are have this character that is the a white ring surrounding black spots. Okay, so this is very clear in Glandon and Pyrenaicus, but also you can see the white ring surrounding the black spots is quite conspicuous in 𝘨𝘰𝘭𝘨𝘶 as well. Now, to tell apart these three species, you look at the blue. The blue is almost gray, very pale in Pyrenaicus. It's gray, darker, I would say, with a wide margin in Glandon. And in 𝘨𝘰𝘭𝘨𝘶, it is restricted to the base of the wings. So this is the different. So the blue is the distinctive character. All the three species have cell spots, although you see here again a specimen that doesn't. This is unusual. Okay, and as you can see in Pyrenaicus, black spots are almost absent, while in 𝘨𝘰𝘭𝘨 they are quite marked. And here you have an intermediate situation in which black spots are tiny, but you can still see them. Okay, so you see the three species can easily be identified in the field. Pyrenaicus and Glandon can live together, but not 𝘨𝘰𝘭𝘨. They have different distributions. So let's have a look to Glandon, the first species. It's present in the Pyrenees and in the Alps. It lives in alpine grasslands and screes. Its larval footprints are, mainly from the genus 𝘧𝘢𝘆𝑒, but also Oxytropis and Soldanella. For example, this 𝘧𝘢𝘆𝑒 vitaliana is one of the typical footprints of the species. And adults fly, mainly during the summer, because this mountain species cannot fly earlier in the season. The males are gray-blue with a white dark margin, as I told you, and the underside have black spots surrounded by a white area, as I told you previously. The next species is 𝘨𝘰𝘭𝘨. This species is restricted to Sierra Nevada, and in southeast Spain, it occupies high altitude. You see very high altitude areas from 2,400 meters to 3,000 meters. And the habitat here is dwarf juniper scrub, and the substrate is always schist screes. The larval footplant is always 𝘧𝘢𝘆𝑒 vitaliana, and adults fly from the end of June to July. They are quite early in the season. Now, I must say that the taxonomic status of this species is under discussion. It is uncertain. Now, quite recent studies support that this species is a subspecies of Glandon. But however, it flies in very different area from Glandon. So this is in the south Glandon. The nearest populations for Glandon are in the Pyrenees. Males are brown in this species with restricted blue, and the underside have the black spots are quite conspicuous. Pyrenaicus is the third species. This is an endemic species from the Pyrenees, the central Pyrenees in Spain and France, and also in the Cantabrian Mountains. It also flies at high altitudes, although not as high as, for example, 𝘨𝘰𝘭𝘨𝘶. Always calcareous grasslands. This is important because Glandon, for example, can use acid rocks like granite or schists, but this species is always to be found in calcareous areas. The food plant is Androsaceae villosa, this very nice white flowering plant, and adults fly from June to August. As I told you, it can also be found in France, in the Pyrenees, but otherwise it is a peninsular endemic species. So the main character here is the pale gray blue of the male's upper side, and also the underside with very reduced or absent black spots. Now let's go to Arythia, and this is the last genus. We are almost got it. Arythia has got 6 species, and we'll be dealing with 3 of them. And I will show, Nithias just for comparison. The Arythia species have, all the species have checkered fringes. Nithias is an exception. Okay, Nithias doesn't have checkered fringes, or the checkered pattern is not so obvious, but all the rest of the species have this feature. And another character that is useful for identification of the Arythia species is that the second post-discal spot of the hindwing underside, this is very large, but you will see it quite clearly in the specimens. This spot here is closer to the base than in other similar species, like for example Polyommatus or whatever. So this is the typical feature for the genus, and you see it also for example, in Nithias. Okay. Other characters have to do with the color of the underside. For example, the color is gray here or brown in Cramera. It is light brown in Montensis, and it's variable in Moronensis. Okay, we will see Moronensis is quite different. But to tell apart these 2 species is quite difficult. You have to rely on the orange spots, but this is very variable, so this is not really a reliable character. Typically, Cramera has a lot of orange spots, well developed, while Montensis doesn't. Okay, and then the apex, this forewing is more pointed in Montensis than in Cramera. You also see that this character is difficult. So reliable identification of these 2 species is difficult, and usually it needs the study of genitalia if you really want to be sure about them. Although Montensis is a mountain species, they sometimes have different clues, but genitalia at the end is the best character. Okay, Moronensis is quite distinct because this wide gap between these post-discal spots and the submarginal markings. Okay, so you see this gap here is wide, and in the other species the gap is very smaller, I would say. Okay, so this is typical for Moronensis. Moronensis is a very variable species, so you must rely on this character to be sure about it. Okay, and of course Nithias is quite different because males are blue in this species, like in other species, like for example 𝘧𝘢𝘅𝘺𝑗𝑒𝑟𝑢s also has blue males. So Nithias is quite easy to tell apart because of the blue males. Now let's have a look to these species, just to have more ideas that make it easy to identify these species. First, Montensis, it lives on mountains, like the name indicates, in open areas, They can have low scrub, but mainly grasslands. And also it's typical of rocky habitats. The larval food plants are always Elaeanthemum and Erodium. For example, Elaeanthemum nummularium, this food plant with yellow flowers, is quite typical. And adults fly from May to September in only one generation. Okay, so that's also quite typical because, as we will see later, Cramera has 2 generations. It lives in southern France, the Pyrenees, and also the Iberian Peninsula. You see, it's widespread in the Iberian Peninsula, mainly in mountains, and it also lives in Morocco, in northern Africa. Now, to identify this species, you have to look at checkered fringes. Males are brown. The underside of this species is light brown, and it's got a pointed apex, as I told you here. This apex of the wings is pointed. The next species is Arythia cramera. Cramera lives in open areas in a wide range of habitats. It's really a very common species in where it lives. Its larval food plants are mainly plants of the Cystacea and Geranacea families. And as I told you, it can have 2 or 3 generations, and in some places you can find it throughout the year. So if you find one or is a species in January, you're almost certainly having a look to Cramera. Okay, the distribution of this species is well, you can find it in the Canary Islands, North Africa, Balearic Islands, Sardinia, and also in the Iberian Peninsula. So in some areas, it flies more or less together with montensis. So in all these areas, you have to have a proper look to the species to be sure about the identification. Usually, Cramera occupies lower altitude habitats, while montensis is at higher altitudes. But in some places they fly together. So this character is again not distinctive. Now, to identify Cramera, look at checkered fringes. The males are brown, and the underside can be brown or gray. Like, for example, here it is brown. While, as I told you, in montensis, it is light brown, so it's quite different. And the last species is Arythia moronensis. This is a species that it is endemic to the Iberian Peninsula. It lives on mountains above 800 meters, particularly rocky areas or screes, but it can also fly in some grasslands, like, for example, in some areas of its distribution range. The larval food plants are several perennial Erodium species, and this is interesting because the larvae, it's also endemic Erodium species. So, for example, this one is an endemic plant. So quite a nice relationship between the butterfly and the plants. And adults fly from May to October, although the maximum is during August. This species is a summer species, I would say. It's endemic to the mountains of Spain, although there is a location in the Pyrenees, here in the French Pyrenees. Okay, so that's the only location outside Spain. Now, to identify this species, you look at first checkered fringes. Males are dark brown, and there's a wide gap between post-discal spots and submarginal spots. So this wide gap is quite distinctive. Okay, so to end up, the conclusion keep an eye on these features I told you about at the beginning of my talk. First, well, of course, these features are important, but as I told you, for most of the species, particularly endemic species, it is very important to know where you are. So the geography is also important. So the features are fringers, whether they are checkered or not. The cell spot on the forewing underside is also important. It can be present or not. Also, the position of the spots in the hindwing underside is important. For example, it's very important to tell apart the Arisia species. Another feature is the present shape and color of the orange lunules. As I told you, you have to look properly at this trait. The shade of blue is important. If the species has blue males, it's important to have a look to this blue to identify this species. And then the last feature is the presence of white triangle or band or this band that is typical of Anomalous Blues on the hindwing underside. So this is the things you have to look at. And of course, I told you a lot of things about the species, but you always look at these characters to be sure about what are you looking at. And then, well, of course, I have to thank this project that has invited me to give this exciting. It's been exciting for me to prepare this talk because it's about species that I love. So it's really nice to. And thank you everybody to be listening to my talk. And of course, you know that this talk has been recorded, so you can have a look to it whenever you like. And really, thank you very much for your attention, and of course, open to any questions.
All right, thank you very much, Miguel. Yeah, we can give some time for some questions. Look, we have one here from Andrew. Returning to Lysandra hispana and Lysandra albicans, can emerge dates help in confirming identity?
Yes, but well, sorry. Yes, because oh, sorry about. Hispana has typically 2 generations, and albicans has typically 1 generation. But there are populations in which this is not true. So yes, in most places you can. This trait is important. So, for example, if you're in Catalonia in May and you find a Lysandra species, then you more or less can be sure. Well, a Lysandra of this group, you can more or less be sure that it will be hispana. But then the second generation is tricky because it's always they always fly in July. So if you find it in July, you're never sure if it's hispana or albicans. So this is an important feature, and thank you for taking this into account. But it's not 100% distinctive, I would say. So you have to be careful about this trait, okay? And again, distribution is probably more important in this case. Okay.
All right. I think there are no more questions, but I can ask something also about these 2 species. The maps you presented, Miguel, from the atlas. So if we are in those regions, either with the distribution in purple or in green, I think you.
Yeah. Sorry,
yes.
Yeah.
So if we are in more like in the Mediterranean coast, or
if you are in the Mediterranean coast, from here, this is Alicante and this is Catalonia. So all this area is Hispana, okay? Mm-hmm. Then if you go further south, you won't find Lysandra species in the coast. So you go to the mountains, and then in the mountains you have to be careful. In Sierra Nevada, it can be Hispana, but in Cazorla, all these sierras here, it can also be Hispana, but in the surroundings it can be albicans. So you have really to have a look to this precise map, because otherwise you cannot be sure about it. Okay, and of course, if you are in the western part of Andalusia, it will again be albicans, and of course in the center of Spain, it will also always be albicans. So, I mean, the map helps, but it is not very useful in the contact areas here in eastern Andalusia, and then in all this area between Cuenca and Teruel and Valencia region. All these areas also quite difficult, I would say.
Yes, and they will be a mix of
yeah, well, they don't fly together, or at least we think they don't fly together. You can never be 100% sure about this. But again, in each place, you have to be careful, because you can have one species or the other if you are in this contact area. Okay.
And Miguel, what is the red dots there in the map?
Oh, these are sorry about this. Yes, these are outliers. So probably wrong records.
Okay.
Yeah. So
thanks.
Yeah, I wouldn't be looking for Lysandra species, for example, here in Sevilla or in Extremadura, but there are records for the species. Okay.
Okay.
Yeah.
Great.
Okay, Cristina, what do you think? Should we? Well, we have given enough time, I think, for any other question. I think people, I think they have, they are still processing some information. I think it was also very, very well explained. And they
very
always,
clear. I was taking notes. I'm very clear on the maps all together, and the putting all the pictures together is easy.
The only thing, it's clear when it is clear, but but the drawback of all these identifications is that in some cases you cannot be 100% sure.
No.
But it's how it is. I mean, some of these groups are evolving at the moment, so it's really difficult to tell them apart. But it's how it is. So when it is easy, it's quite clear. You have clear-cut features, but when the species are difficult because they are just evolving at this time, then it's difficult, and you have to rely on DNA studies. In fact, okay.
Absolutely.
Okay. Thank you so much, Miguel.
Thank you very much, Miguel.
Okay, thank you for attending, and thank you for the questions.
That was a brilliant talk.